On the evening of the 28th of February 1953, two scientists, Francis Crick and James Watson, walked into the Eagle pub to celebrate a good day's work at the nearby University of Cambridge Cavendish Laboratory. Crick boldly announced that they had just discovered the secret of life. As hyperbolic as it sounded, a few months later, the pair published a paper in the scientific journal Nature, where they revealed that they had discovered the structure of DNA, the stuff of life, the iconic double helix. In their words, this structure has novel features which are of considerable biological interest. It explained how genetic information is stored, copied, and passed on through the generations. The same journal also contained papers by Maurice Wilkins and Rosalind Franklin, who had been responsible for the research and the defining image that enabled Crick and Watson to create their structural model. The elucidation of the structure of DNA was a turning point in molecular biology. It started a scientific revolution and ushered in the era of genetic and genomic research that would change medicine as we know it.
So to build a human body, you need to know what proteins to use, you need to know what processes to activate and when and where in the body. And the DNA both gives the information to build the proteins and contains the information, the programming language, if you like, of when and where to switch those proteins on. So DNA is a very simple molecule in some ways. It consists of a string of bases, and we represent those bases by 4 letters, A, C, G, and T.
Professor Julian Parkhill works at the Wellcome Trust Genome campus outside Cambridge. Scientists here have always been at the forefront of genomic research, and they were part of the global team that undertook the biological equivalent of travelling to the moon, the Human Genome Project. Work started on the project in 1990, and it was one of the most ambitious scientific ventures in history.
The human genome has around 3 billion base pairs. There are 2 copies of every gene, so in total around 6 billion base pairs, but most of those are identical. So the original project aimed to sequence that haploid genome, the single copy, the 3 billion base pairs. So the Human Genome Project was a massive undertaking. It involved very large numbers of different people from different countries, different institutes, working at different times. So it's difficult to be exact about the numbers, but people estimated that it cost probably around $3 billion. It involved at one stage or other at least 5,000 people from 20 different institutes around the world. We produced a third of the finished sequence, which is the largest single contribution from any institute around the world.
The Human Genome Project was completed in April 2003. The reference genome determined the exact order of base pairs in a human being and is presented as page after page of text with just 4 letters, A, C, G, and T. Each gene appears as bold text on the page.
They have printed it out in a set of books, and it's a bookshelf around 3 feet wide and 6 feet high, and it contains 40 or 50 books of a very large hardback size, so it's a substantial amount of text.
Today, the scientists working with the human genome in laboratories around the world are fundamentally changing medicine as we know it.
My name is Eli Papa-Emmanuel, and I'm a postdoctoral research fellow at the Cancer Genome Project at the Wellcome Trust Sanger Institute. So the Human Genome Project was completed around the time that I was finishing school, and it was a very exciting time, and I came to the UK to pursue studies in human genetics, and that then has taken me into cancer research for my PhD and postdoctoral research. The completion of the human reference sequence has been fundamental in strengthening our understanding around both how the genome sequence is organized, but most importantly, how this correlates to disease.
Once the reference human genome was completed, it became possible to study groups of patients who shared the same disease to look for shared genetic markers or gene mutations.
Specifically, we now know that the human genome contains approximately 23,000 genes, of which 5,000 with certainty and more likely up to 10,000 of those genes have been correlated with common diseases such as cardiovascular disease, arthritis, as well as more rare diseases and developmental disorders such as autism. Our understanding of cancer has been revolutionized through the attainment of the reference genome sequence, with at least 400 genes known to be implicated in cancer today. So, by having a very well annotated list of correlation of gene mutations with different disease entities, and the great opportunity of having now these technologies incorporated within clinical laboratories, it is now possible to test a patient's DNA alongside all the routine diagnostic tests for those common genetic markers that can inform us not only on the specific disease entity that a patient may be associated with, but also provide quite a lot of important insight on potential new therapies that would be effective for that specific patient, taking into account their genomic profile.
Set. I understand you've got some family in the
While there's still a long way to go in translating research into clinical practice, progress so far has been very promising.
95% of the time.
In the same way where we currently think a blood test as routine, in the future a DNA test should inform what the patient suffers from, what the most effective treatment is, potential adverse reactions to specific treatments or combination of treatments that one should be mindful of, and expected outcomes, so that patients are not overtreated or undertreated and rather receive the best possible care that they can. And all of this with a very simple and easy to conduct as well as interpret DNA test that's going to be routine across clinics and why not GP practices.
We made it from the discovery of DNA's structure to the completion of the Human Genome Project in just 50 years. We now live in a world of genomic medicine, and it's a world of endless possibilities.